Hello everyone, and welcome back to the Unlearn channel. We often see snails, but have you ever wondered just how intelligent this tiny creature is? Today, we're diving into the mysterious world of the snail brain. Get ready to be surprised by the unique facts we're about to uncover. This video will prove that size isn't everything. Even a creature as small as a snail possesses remarkable intelligence. Make sure you don't miss a single segment, but first, hit the like and subscribe buttons to support our unique research. We'll cover everything from their long-term memory to how this miniature brain holds the keys to human neurology. We start by getting to know its unique brain structure. Let's virtually dissect this snail. Let's start with the basics. A snail's brain isn't a solid mass like a mammal's. They possess clusters of nerve cells called ganglia. These ganglia are located surrounding the esophagus, forming a nerve ring. Just imagine a neural necklace around their neck. One of the reasons snails are so important to science is their neurons. Some of their nerve cells are much larger than human cells. These ganglia receive and relay signals. It's a miniature control center managing everything from movement to feeding. This difference in neuron size makes it easier for scientists to study how electrical signals and synaptic connections work. The ganglia are divided into several parts. There are the cerebral ganglia, which handle the senses in the head, and the pedal ganglia, which control movement. Take the sea snail, Aplysia, for example. Its graceful movements are controlled by its pedal ganglia with astonishing precision. There are also the visceral ganglia, which regulate internal organs and vital functions like the heart and breathing. This simple, though small, nervous system operates highly efficiently for the essential tasks the snail must perform. When a snail's tentacle touches something, the signal zips to the cerebral ganglia, and a quick response is processed in mere milliseconds. Imagine the complexity of managing organs with only a few thousand neurons. This demonstrates the amazing efficiency of nature's design. To give you perspective, the entire snail brain is only about the size of a grain of sand. Yet its capabilities far exceed its size. These neural pathways ensure sensory information enters, is processed in the ganglia, and motor commands are sent out. A neatly structured system. Snails don't just see, they also smell and feel the environment through chemical receptors. The ganglia process this complex input. The most fascinating part is how these cells know where to connect, a process called axon guidance. Interspersed among the neurons are glial cells. They may not fire signals, but they are crucial for supporting neural health. Communication between neurons happens via neurotransmitters, like chemical messengers released into the synaptic cleft. Every neuron has a nucleus that contains the cell's blueprint for life, controlling all biological processes from its center. The snail's seemingly slow movement actually requires constant neural coordination to control the slime and foot muscles. Snails also have a balance organ called the statocyst. This sends signals to the ganglia to maintain orientation when crawling up or down. When we isolate the ganglia, we can observe their subtle pulsing and electrical activity. Although a snail's vision is simple, the ganglia process what they see at the tip of their tentacles, distinguishing between light and darkness. The speed of a snail's tentacle withdrawal reflex is proof that its nervous system operates with a rapid response to danger. Unique fact: garden snails' brains contain only about 11 to 20,000 neurons. Compare that to the human brain, which has 86 billion. Now we get to the most surprising part: snail memory. Yes, these little creatures can learn and remember. They exhibit habituation. If you repeatedly touch them without harm, they learn to ignore the touch. Conversely, there is sensitization. If the touch is followed by danger, their response will be stronger and last longer. They learn to be alert. This learning changes the proteins within the neurons. This protein change is the physical trace of their long-term memory. Short-term memory occurs because of repeated signals, making the synapses temporarily more responsive. This habituation process is highly efficient. They save energy by not reacting to every irrelevant stimulus. Snails are even capable of associative learning, linking a specific smell to a bad taste or dangerous experience. Key neurons like L7 in aplesia become the center for synaptic strengthening when a memory is formed. To form a permanent memory, neurons must create new proteins and grow new synaptic connections. Maze studies show that snails learn from their mistakes. They are able to recall successful routes they have taken. Short-term memory uses existing signals, while long-term memory physically reinforces the neural pathways. In the wild, this means they can remember where the best food sources or hiding spots are, even after several days. For memory to persist, genes within the neurons must be expressed, creating new proteins that alter the neural structure. If they've eaten something that made them sick, that smell will be associated with a bad feeling for a long time. They possess an amazing homing ability, returning to the same resting or hiding spot after traveling far. The process of recalling memory involves reactivating the same neural circuit that was formed during the learning process. This phenomenon is called synaptic plasticity. Connections between neurons can strengthen or weaken, much like a muscle being trained. Even while resting, the snail's ganglia continue to process and consolidate the information they received throughout the day. Key proteins like CREB and MAPK are the unsung heroes behind the scenes, responsible for memory stability. When faced with a new object, their tiny brain works hard, deciding whether the object is important or threatening. Snails can be trained using classical conditioning, just like Pavlov's experiments, proving a profound capacity for learning. After the sensitization process, the release of neurotransmitters is enhanced, ensuring a faster and stronger response in the future. Every strong memory forms a new neural pathway, literally changing the circuit map within the snail's ganglia. The trained avoidance behavior of the snail becomes highly coordinated, no longer a spontaneous reaction, but a learned response. The neural network before learning appears messy, but after learning, the circuit becomes structured and efficient. Environmental memory helps snails survive, ensuring they always know the best place to avoid predators or extreme conditions. Calcium ions entering through special channels are the main trigger for the chemical signals that lead to memory formation. Their sense of sight is limited, but enough to process vital information that is then stored in the ganglia memory. The snail's ganglia quickly interpret signals from taste sensors to differentiate between food and potential toxins. Some species of sea snails have been proven to maintain associative memory for at least 5 days, a remarkable achievement for such a small brain. This communication process occurs within a dense network, proving that even with few neurons, the speed of information is maintained. For long-term memory, new proteins must be synthesized. Without this process, memory will only last temporarily. In the context of the sea, memory is crucial. Snails that remember the location of predators have a much higher chance of survival. Long-term potentiation is the lasting strengthening of synapses the basic mechanism of learning and memory in almost all animals. On the other hand, there is synaptic depression, the weakening of connections. This is the brain's way of forgetting or ignoring unimportant information. Although they don't have ears, the snail's nervous system is highly sensitive to vibrations and loud noises in the environment. Some research also suggests that neurogenesis, the growth of new neurons, occurs in adult snail ganglia. Specific proteins ensure that long-term memory traces are maintained by breaking down unnecessary proteins. Successfully completing a maze is a clear benchmark that the snail is not just moving randomly, but using spatial memory. Every important experience literally etches deeper pathways into their neural circuits, facilitating recall in the future. Snails learn which surfaces are easiest and safest to climb, storing this tactile memory in their pedal ganglia. The adenylate cyclase enzyme activated by serotonin is the key biochemical factor in the snail's synaptic strengthening process. Interestingly, there is evidence that snails can learn from other snails, a kind of primitive social learning. Even in seeking a mate, the snail's ganglia process chemical pheromone signals, guiding complex reproductive behavior. All these mechanisms, from habituation to LTP, are clear evidence that snails possess a significant level of intelligence. They are capable of fine distinction between two very similar stimuli, demonstrating discriminative learning ability. If one path is dangerous, this small brain is capable of creating an alternative neural route to avoid that danger. So, when you see a snail avoiding a leaf that was previously poisonous, it's not a coincidence. It's learning stored in its brain. Why are scientists so obsessed with this simple snail brain? The answer lies in their large neurons. The snail's large neurons allow researchers to isolate and manipulate individual cells, which is near impossible in the human brain. Techniques like patch clamping are performed on snail neurons to measure their electrical activity with high precision. Understanding how serotonin changes the snail's synapses has given us key insights into how memory works at a molecular level. Snail research has paved the way for biohybrids, combining living nerve cells with computer hardware. The snail's simple memory mechanisms serve as a model for understanding and perhaps treating neurodegenerative diseases like Alzheimer's. Researchers globally continue to discuss and visualize snail brain models to crack the memory code. The basic principles of memory discovered in snails are universally applicable to almost all living creatures, including us. The journey through the snail's neural pathways is a fundamental voyage into the biology of memory itself. Even robotics is inspired. Scientists study the snail's motor control to create more efficient robots. In fact, key research on aplysia by Eric Kandel won a Nobel Prize, showing just how important this snail is. Snail genetics are also studied to understand how the DNA code directs memory formation. Through genetic engineering, researchers can see which neurons are active during the learning process in real time. The snail is a near-perfect biological model for studying a complex yet accessible nervous system. Snail neurons can live and function normally in a petri dish, allowing for profound experiments without involving the entire organism. In the laboratory, the snail is becoming an unexpected hero in our quest to understand the mysteries of the mind. The snail's simple neural circuits are being converted into mathematical models to program intelligent machines. They also help us understand why we forget, the long-term depression process that weakens connections. Research on snails continues to push the boundaries of neuroscience toward the future. Virtual simulations of the snail brain help students and researchers understand its functionality. Every detail of the snail, from its shell to its ganglia, is a valuable source of information. Snails are used to test how the nervous system responds to physical stimuli from the environment. These sophisticated research systems ensure that every experiment is conducted with maximum control. Detailed understanding of the pedal ganglia gives us the key to controlling muscle movement in biological systems. Snails are even used to study proteins like prion, which are involved in memory, not just disease. The snail ganglia show an efficient mechanism for nerve cell repair after damage, attracting the attention of regenerative scientists. Hormones controlling the snail's reproductive behavior are also studied as a model of a simple endocrine system. Their nervous system is the inspiration for designing more efficient computing circuits and algorithms. The snail's slow, stable movement inspires the design of robots and drones that require precise crawling. Studying how the ganglia develop from the larval stage gives us clues about how the brain grows and organizes itself. Even the simple sleep patterns of snails provide clues about the role of sleep in memory consolidation. Snails also exhibit an immune response controlled by the ganglia, linking the nervous system and body defense. This is the bridge between the primitive biological world and advanced technology, the snail as the key to understanding. Never underestimate the potential of the simplest system to reveal the most profound truths. Every discovery from the snail brain is a gift to global neuroscience advancement. Essentially, this small snail brain is the key to unlocking the secrets of how memory and learning work, even in us humans. Which fact surprised you the most? Was it the 5-day memory or its large neurons? Write it in the comments below. If you enjoyed this unique facts video, don't forget to subscribe and hit the notification bell so you don't miss our next video.